Welcome to today's Explainer. I am absolutely thrilled to jump into this one with you, because today we're exploring a total paradigm shift in modern medicine, targeted protein degradation. For decades, drug discovery has essentially relied on one very specific strategy. You find a lock on a disease-causing protein, and you shove a chemical key into it to shut it down. But today, we're talking about a totally different approach. Instead of just locking the door, what if we hired the ultimate cellular demolition crew to remove the door entirely? We are diving into exactly how scientists are now hijacking the cell's natural garbage disposal system to cure diseases.
Here's our roadmap for today. We'll start with proteostasis and undruggable targets. Check out the Ubiquitin-Proteasome System, see how PROTACs hijack it, trace their clinical timeline, jump into molecular glues, and then wrap it all up with a recap and some exam prep.
All right, Part 1, Proteostasis and Undruggable Targets.
So, before we can talk about the cure, we've got to establish the stakes here, right? Why do we even need to destroy proteins in the first place? Well, when the cell's delicate balance of creating and destroying proteins fails, the consequences are severe. Misfolded proteins can aggregate and cause Alzheimer's or Parkinson's, while overexpressed proteins, like MYC, can drive relentless cancer growth. And the sheer scale of the problem we've faced in trying to drug these diseases is honestly staggering. For years, traditional small molecule drugs required a deep, active pocket to bind to. Think of kinase inhibitors like imatinib. But those druggable targets, they only make up about 15 to 20% of the human proteome. The vast majority, roughly 80 to 85% of our proteins, are considered completely undruggable. These are things like transcription factors or scaffold proteins that have flat, featureless surfaces. You just literally cannot get a traditional drug to stick to them.
It's all about this really delicate balance. Think about the natural life cycle of a protein. It's synthesized, it folds, and if things go wrong, it misfolds. When a protein misfolds, it can unfortunately go off pathway and aggregate into toxic oligomers or amyloid fibrils. Under normal, healthy conditions, the cell balances this out by sending those damaged goods to the degradation pathways, either the Ubiquitin-Proteasome System or the lysosomal pathway. Targeted protein degradation is essentially our modern therapeutic strategy to step in and forcefully restore this balance when the natural clearance network just fails us.
Moving on to Section 2, the Ubiquitin-Proteasome System.
If the lysosome is the cell's bulk garbage disposal, the Ubiquitin-Proteasome System, or the UPS, is the cell's precision shredder. It's designed specifically for short-lived, soluble, misfolded proteins. But to get a protein shredded, you have to tag it first, and this happens through a highly specific three-step mail delivery cascade. Step 1 is the E1 activating enzyme. There are only about 2 of these in humans, and they act as the gatekeepers. Think of E1 as loading and applying the postage stamp. Step 2 is the E2 conjugating enzyme, with roughly 40 variations in humans. E2 is basically your mail carrier. And finally, Step 3 is the E3 ligase. With over 600 E3 ligases in the human body, these are the intricate addressing modules that ensure the envelope goes to the exact right target for destruction.
Now, the crucial thing here is how energy actually drives this entire cascade. In Step 1, E1 uses ATP to form a high-energy thioester bond with a ubiquitin molecule. This energy investment is irreversible, and it essentially powers the whole process. The ubiquitin is then passed to E2, and finally, E3 brings the target protein close enough to transfer that ubiquitin tag right onto it. And here is a vital detail. It's not just any tag. Specifically, a chain of K48-linked polyubiquitin is the universal destroy me signal.
Okay, so once that K48-linked polyubiquitin tag is securely applied, the protein is dragged over to the 26S proteasome. I absolutely love the structure of this molecular machine. It has 2 main parts. First, you've got the 19S regulatory cap on the top and bottom. Think of the 19S as the paper feeder. It recognizes the ubiquitin tag, rips it off so it can be recycled, unfolds the protein using ATP, and then feeds it straight down into the 20S catalytic core. That 20S core is the actual shredder. It's a barrel-shaped chamber that ruthlessly chops the target protein into tiny peptides, usually between 3 and 25 amino acids long.
Which brings us to Section 3, PROTACs and hijacking the UPS.
Back in 2001, scientists had this incredibly elegant idea. They realized, hey, since 85% of proteins don't have active sites that we can inhibit, what if we just built a molecule to drag those disease-causing proteins directly to an E3 ligase? We don't actually need to inhibit them, we just need to hijack the UPS to destroy them. To pull off this hijacking, you need a very specific blueprint. A PROTAC is a bifunctional molecule made of 3 parts. On one end, you have the warhead, a ligand that binds your target protein of interest.
On the other end, you have the E3 ligand, which recruits a specific ligase, like CRBN or VHL. And bridging them together is the linker, often made of PEG or alkyl chains. This linker isn't just dead weight, by the way. Its exact geometry is absolutely critical for pulling those two massive proteins together perfectly.
When those three parts do their job perfectly, they form what's known as the ternary complex. This is the very heart of a PROTAC's action. And what's absolutely magical about this formation is something called cooperative binding. By bringing the target and the E3 ligase together, they actually increase each other's affinity. For example, a PROTAC might bind its target with a relatively weak KD of 100 nanomolar on its own. But when the E3 ligase is present, they lock together so incredibly well that the binding affinity jumps to a tight 10 nanomolar.
This literally changes the entire math of pharmacology. Let's look at this comparison. Traditional inhibitors are occupancy-driven. You need a 1-to-1 stoichiometric ratio to keep the drug bound to the target constantly. It's just like putting a lock on a door. If the lock falls off, the door opens again. PROTACs, however, are event-driven. They act catalytically. One single PROTAC molecule can force the tagging of a target, release it to be destroyed, and then move right on to tag 100 more targets. It's substoichiometric. It's that demolition crew coming in and removing the doors entirely.
All right, Section 4, Clinical Timeline of PROTACs. If you track this timeline, the exponential growth is just staggering. In 2001, Sakamoto, Cruz, and Deshays published the very first PROTAC concept using a peptide. By 2008, we had the first small molecule PROTACs. But 2019 was really the watershed moment. That's when ARV-110 and ARV-471 became the first PROTACs to enter Phase 1 human clinical trials. And now, moving past 2022, we have multiple PROTACs actively in Phase 2 and Phase 3 trials, and we're expanding way beyond cancer into autoimmune and neurodegenerative diseases.
Looking at the actual clinical pipeline data reveals this real-world impact. Take ARV-110, right at the top there. It targets the androgen receptor for metastatic castration-resistant prostate cancer, hijacking the CRBN E3 ligase. It reached Phase 2-3 trials because it successfully degraded AR in patients who had literally failed all standard therapies. We figured out how to shred the exact protein driving their cancer. You can also see ARV-471 doing the exact same thing for the estrogen receptor in breast cancer.
Moving on to Section 5, Molecular Glues and Thalidomide.
First, let's clearly differentiate these two strategies. PROTACs are bifunctional, meaning they have two distinct arms, and they are relatively large, weighing in at about 800 to 1200 Daltons. Molecular glues, on the other hand, are monovalent. They are tiny, single molecules, only 300 to 500 Daltons. Instead of acting as a long bridge, a molecular glue wedges itself right into the E3 ligase and subtly reshapes its surface, forcing it to accidentally lock onto a target it would normally just ignore.
Now, you probably know the tragedy of thalidomide. Back in the late 1950s, it was prescribed to pregnant women for morning sickness, resulting in thousands of devastating birth defects. But decades later, thalidomide showed these incredible anti-cancer properties. It actually wasn't until around 2010 to 2014 that scientists realized why. The old model assumed it had multiple random targets, but the current model shows thalidomide contains a conserved glutamide ring. This ring binds directly to the CRBN E3 ligase, reshaping it so it grabs onto neo-substrates like the transcription factors IKZF1 and IKZF3. Without even knowing it, we had accidentally discovered a molecular glue.
Okay, finally, Section 6, Lecture Recap and Exam Prep.
Let's lock in the core takeaways from everything we've explored today. Number 1, K48-linked polyubiquitin is the specific signal for proteasomal degradation. 2, degraders utilize event-driven pharmacology, making them catalytic and substoichiometric. 3, remember the numbers. Humans have about 2 E1s, 40 E2s, and over 600 E3 ligases providing that exquisite specificity. 4, the ternary complex is the absolute heart of degradation. And 5, for molecular glues like imids, that conserved glutatamiide ring is structurally mandatory to bind CRBN.
Let's wrap up this explainer with a provocative question for you. It's called the hook effect. Why does an overly high concentration of a PROTAC actually decrease target degradation? It seems totally counterintuitive, right? Well, the answer is that too much PROTAC saturates the target proteins and the E3 ligases separately. Instead of forming the required ternary complex, you just get a bunch of useless isolated binary complexes. The whole system just gets clogged up. It really goes to show, sometimes in pharmacology and especially in cellular demolition, more isn't always better. Precision is everything. If we can successfully hijack the cell's garbage disposal to cure the undruggable diseases of today, it leaves us with one massive question. What other natural cellular machines will we learn to hijack tomorrow? Thanks so much for exploring with me today. Stay curious.